The next speaker is Dr. Swagatika Sahoo. Dr. Swagatika Sahoo is a senior scientist currently at the Vantage Research in Chennai. Briefly, a bit about Swagatika. Swagatika did her PhD with Professor Ines Thiele at University of Luxembourg. As you know, Professor Ines Thiele and Professor Palsson are pioneers in genome scale reconstruction and modeling of human metabolism. Swagatika was also involved, I understand, in the work on Recon 3D. So she is an expert on genome scale modeling of human metabolism. After her PhD, she spent quite a bit of time as a DST Inspire faculty and then a senior scientist at IIT Madras. And recently, she has moved to Vantage Research to work more in the industrial space. So it's wonderful to have Swagatika, and we're looking forward to your talk.
Hi, good morning, everyone. Oh, I see my Vantage folks. Hi. Okay, so thank you, Arijit, for inviting me and giving me this opportunity to talk about the human metabolic network. And what I'll be talking today is how we can use this network for different disease research context and various other fields of medical science. So the outline of the talk is we'll discuss a bit about the network or the foundations for the human metabolic network, what is the paradigm and what is the basic use currently. Then we'll move on to a test case of a disease called retinoblastoma, and then finally the very recent, the whole body metabolic model, which is organized as organ resolved gender specific whole body models. Okay, so I'll start with the central dogma of life, which is basically the flow of information, that is nothing but translation of the language of nucleic acids to the language of proteins, and finally the metabolic transformation, where you have the conversion of metabolites to different products, and they do also carry a lot of information. So this is the information flow, and corresponding to this, what we have are the different layers of data, and as we call them now are the different omics. And the omics goes beyond this. I just noted the most highly used omics data currently. Genomics, we have now started out the Indian version of the cancer data, which is called the ICGA, which is completely the genomic information of the different cancer types across Indian patients. Then we have with the level of the mRNA, we have the corresponding data, which is called the transcriptomics. The proteomes, basically the entire protein information that is there within a cell. And then, of course, the metabolomic level, which is the concentration of metabolites under different conditions. So when you have this humongous level of data, what do we use this for? So that's where the next usage comes, which is basically the systems biology paradigm, which is we integrate all of these data types onto a model of our choice. And the model that I will be talking about, it is called the human metabolic model, and which applies on the principle of steady state modeling. What we do is we integrate different kinds of data types. These are the ones that we just discussed, but we can go beyond them as well. And then we build the network of our choice, and we use different query or model analysis tools to answer a lot of biologically relevant questions. Most of the time, the omics data, if you're using it for disease context, has been used for prediction of different biomarkers, as well as to understand the underlying pathology, what exactly is going wrong at the proteome level or the metabolite level or the genomic level, depending upon the data that you're using. So, but the crux of this entire effort is integration. So you integrate different kinds of data types onto one single model, in this case human metabolic model that I'll be discussing, or it could be any different kinds of networks. Okay, so the context that I'm talking is the biochemical reaction network, or at the level of metabolites, the lowest layer, which is the metabolomics data level. Okay, so what do I mean? We are talking about this particular system, which is phosphorylation of glucose. that is glucose from the outside of the system is getting inside and then getting phosphorylated. The corresponding biochemical reaction network will then capture this particular information in detail. We go on to the level of the metabolite, how are they interacting, as well as the typical subsystem. So when we talk about the different subsystem, what I'm meaning is the intracellular organelles. That's the level we try to include in our reconstruction. So typically the same system again, the metabolic network will then comprise of the different metabolites and then the reactions, of course. And then finally, one of the most important part that it will then have is the genes, which will then, the genes will encode the proteins / enzymes. Enzymes will then catalyze these reactions. So all of this information are then encoded within this particular network, and you can talk it as the nodes and edges, which will be metabolites and the reactions. It could be also built at a level of genetic gene regulatory network, protein-protein interaction network. There are different kinds of networks that are out there, and I think most of the other speakers will talk about them, but this particular talk is focused on the metabolic network. So this is the most comprehensive, the most recent human metabolic network. What we did was we tried to capture the data as well as the literature information that is out there on human biochemistry into a readily computable format and then use it for various purposes. So this is the most comprehensive one. This got published in 2018. We call this the Recon 3D version. We have previous versions of it. The first version was in 2007, which was Recon 1. And then came around 2013, that was Recon 2. And then we have now Recon 3D. I think after, now the efforts are being put in to have the fourth version, which is mostly catered towards using this particular network to study the inherited metabolic diseases. But this is more of a generic network. It captures any kind of cell that you will observe in the human body. And the building was done both from the different kinds of omics data as well as lot of literature information, lot of physiological information, and protein structural information as well was encoded into this particular network. I will give one example because majority of the information in the third version came from this particular chunk, which is the metabolomic data. We used at least 10 different sets of whole cell metabolomics data, and we ended up having around close to 14,000 reactions and 4,000 metabolites. So how do we go about using or integrating the different metabolomics data in this case is we start out with the metabolite of your interest in your data set that you have got. You curate the biochemical literature on the different kinds of reactions that this particular metabolite is used as well as synthesized, and then you end up having the typical pathway. So from single reaction you end up having a different pathway. You go on doing that for a lot of metabolites of interest, as well as you can use a lot of databases also that contain this particular information. So from pathway you then finally end up having this particular huge network, which is, as I said, it has around 13,000 reactions. So what we did have with respect to the previous version Recon 2, we had a lot of new reactions, given that most of the metabolites that we added up were new. Majority of them actually came from lipid metabolism. Lipid as well, it is still in the stage of the lipid metabolism, and lipidomics area is still in the research and advancement stage, but still we were able to capture a lot of it. And then we had new metabolites and new genes which were not captured in the previous version. So once you have this entire information there, you do a lot of this quality control, quality assessment tests to see whether your network is able to perform all the functions for which system that you're basically building for. In this case, because we were building for human, we had a lot of these metabolic objectives that a human body should be able to perform. So these are basically metabolic objectives of each kind of different cells, converted them into mathematical equations and then optimize them for. So that is where we now go into the stage of from reconstruction to a model where you're actually testing the capabilities of the system. So what you do is you convert the system into a matrix where you have rows and columns. Your rows are taking up the metabolite space and the columns are your reaction space. And then you define your system's boundaries. This is again where you have to have a bit of domain knowledge, where you have to say these are my sets of inputs, and sometimes your outputs are defined depending upon the data. And then you formulate your objectives. Most of the objectives that we used were human specific objectives, and that's what we tested. And then of course we also had to take care of a lot of constraints which were physiologically relevant. So once you have this thing defined, this is a typical protocol that any genome scale metabolic model that you are building for have to have these kind of built-in functions. Okay. So the method that we used for testing the model objective functions, whether they carry active flux. Now I'm using a new term here. So flux, what I mean is simply the rate of the reaction. Okay, whether my reactions are active in the model or they're just simply lying there. So that's what you test for. You ensure that at least 80 to 85% of your model is active. It is carrying a flux. Usually what we use is the linear optimization toolbox, and this particular method, which is called the flux balance analysis, is not new. This existed. This was first used for microbial systems, where the objective was to come up with certain industrially relevant products using microbes. That's why they use the flux balance analysis. We use the same technique for human system. Okay, so this method is pretty old and tested, but we have come up with newer versions of flux balance analysis. Now when you look into this toolbox, it has around 110 or 112 model testing methods. Okay, so you can just choose which particular method or group of methods that you are interested in. And of course, the question that you're trying to answer. Okay, so then once you have the objectives defined, your constraints are defined, your model is running fine, what do you do next is you try to add in the biological relevance, because all of this is meant for the human system. You have to answer certain real world questions. So I'll just give one example of an objective function. How did we convert biology into a mathematically relevant function? Okay, so let's say our objective here is energy production of glucose, which is very common function of the human body. We eat to sustain. We use glucose to derive energy, and one of the basic pathway under which glucose is oxidized and converted to energy is glycolysis, and that's what we test for. We give in the constraints that you have to have glucose in the medium, and of course you have to have unlimited supply for oxygen because it's a aerobic oxidation. So what we do ensure during this particular exercise is this entire pathway of glucose oxidation is first present in the cell. Most of the times, because you're using omics data, which is only a snapshot of that particular condition, your data will capture only a chunk of this particular information. It could be only under this or only this particular thing. So you have to read a lot of literature to ensure that this particular entire pathway is present in the system. There are certain tools and algorithms out there which can also do it automatically for you, but then you have to have certain biologically relevant functionalities associated with these functions. So that's all the message that I want to give in this particular slide was we tested a lot of these functions, and also it mentions 431, but we actually went to close to 450 functions. And yes, of course, these are single optimization, one at a time. There are again objectives and tools where you can go for multi-level optimizations as well. Okay, here. What we also ended up having is these organ-specific metabolic objectives. Because we were building a model which was very generic for the human body, the question was, can we now also capture certain organ specific or cell specific functions. Okay, so we ended up having close to 100 such functions, and we tested, and it all came to be true for this particular model. Now that we have this model, this particular model has been used number of times for different questions, majorly in the medical field. The one that I just talked about, and I also give you another example, is how do we use data. It could be omics, it could be different kinds of clinical data also. How do you map that onto your model to answer certain disease specific question? You can also use this particular model to answer this particular cell-cell interaction. How are the organs within the body talking to one another at a metabolic level, or even at a genetic level? You can do that with this particular model. And here comes another most hot topic currently, is host microbe interaction. You can integrate this particular human model with a number of microbial models out there to have a host and your microbe interaction, because majority of the diseases, typically metabolic diseases that we see today, we see a lot of association with the gut microbes. So how is it that the gut microbes are interacting with the host cell, leading to a disease, or could even be tuned to cure the disease? So all of these questions can be answered. And then of course there are various other, the typical biomarker and drug discovery processes also can be used for this particular model. One example that I'll be using now is a cancer, and this was done in collaboration with Shankar Netralia here. They were the ones who provided the typical omics data for retinoblastoma. And retinoblastoma, it's a childhood disease. It is caused, there are different stages of the disease, and the abundance in typical the Indian population is slightly higher compared to the Western one. Okay, so the question that we wanted to answer, can we now use this particular model to understand the biochemical basis of this particular cancer type and hopefully use it to predict certain novel therapeutic strategies? So yeah, these were the objectives that I just talked about. So we started out with, of course, the human metabolic network. We had the data, which was one constraint, and then we used different model generation algorithm. There are these number of model generation or model extraction algorithm depending upon the data type that you are putting it in. Okay, so you end up with a primary reaction network. However, as I said, data will capture only one particular condition under which the experiment was set up. Your basic aim here is to capture the totality of that particular condition. Okay, so you do end up reading again a lot of literature. You test your model against a lot of objectives for which the system was built for. And then we ended up having around close to 55 retina specific objectives. Asked our model, are you able to perform this reasonably well? Did a lot of these iterations to actually have activity for these particular objectives, and all of these models were average around 5,000 reactions. Okay, this is what I show on the x-axis is the activity of those objectives across the different cancer types or the cancer models, and here are your y-axis are the objectives. Okay. So what we saw when you compare the cancer versus the normal condition, we saw that the cancer was preferentially using amino acids for energy versus the carbohydrates, which were typically being used by the normal retina or the normal healthy cell. Okay, so why are we interested in these kind of energy precursors? Because these are basically the answers. Can we now switch the amino acid usage to carbohydrate in order to get rid of the cancer? That's the reason we are interested in the pathology of the disease. Then what we saw is a very interesting phenomenon that when you come to a metabolite which is called cholesterol, cholesterol is there across every cell membrane. It is a very important metabolite for proper maintenance of the cell type. What we saw is the cancer, typically the retinoblastoma cancers, they were underutilizing the cholesterol. They were kind of saving the cholesterol for something in the cell. And then we asked the model, why is this happening? It shows that when you go for cholesterol synthesis, there is a redox potential, which is the NADPH in this particular case. It was kind of maintaining or preserving this NADPH so that it will mimic that nothing is wrong in the cell, everything is all right, and your protection functions are then not kicked in. So if anything goes wrong, there is something called the protection functions in the cell which will then be kicked in to fight this particular condition. But it was underutilizing cholesterol to preserve those extra metabolites, in this case redox potentials, to not trigger the protection mechanisms of the cell. So that is what was happening in the cancer type. Another important feature that we saw across the different cancer types was these different models were very specific in synthesis of fatty acids. This we went into literature and then we saw that this is actually a hallmark for the different stages of the cancer. The different stages that I showed previously, they were very specific to the different fatty acids, and the models are very nicely capturing this. And what we also did, we were able to so all of this well and good, but you have to challenge your model with the real world scenario. We then used the experimental validation support from Dr. Miles in Ohio, and she very gladly did the validation for our secretion pattern of the cell, and it turns out that 60% of what the model was predicting was actually true. So reasonably okay models we were happy to get with. So this was the cancer test and one of the disease area that you can use the model for. Another study that I would like to discuss today is the recently published organ resolved whole body metabolic models. This is the very first time we were, well, not very first time, but the interaction among typically the metabolic and the physiological interaction among the different organs of the human body, we were able to do it on the whole body level, capturing the interactions across 32 different human organs. So what we did, we started out again with Recon. We used a lot of data, of course, and then of course we used a lot of literature information to put across the different adult organs, blood cell types, excuse me, and also gender specific organs. And then we used one or the other model building algorithm. As I said, there are at least for this particular tool works that we consistently use, it has inbuilt 6 different model building algorithms. So you are free to choose whichever you want. So, but again, this is the most important part. You have to test your models for the functions for which the system is being built. So we had around more than 100 iterations to finally achieve to this particular finished models, and what we got at the end are the molecular level organ resolved gender specific metabolic models of the whole human body. For the male we had around 28 organs depending upon the data for which these exist for the different organ types, and for the female we had close to 30 organ types. Where is Arijit? 5 minutes, huh? Okay, fine. So yeah, so this is the whole body model. As you can see, we had established the models for the different organs. This is nothing new. Many people are doing it, but what we did, something very first time here, is we established these 13 connections, and these connections are the typical biofluids that you see interacting and connecting these organ types, both for the case of adult organs, gender specific organs, and the blood cell types. So that is what we established here, and that is something we benchmark this model, that we are now able to capture not only the organs but their internal connections, and now we are at a level to actually capture the biofluid specific biomarkers for the various diseases. So yeah, of course, these are pretty huge models, because you can imagine if one particular, the retinoblastoma for which I built the retina model, which had close to 5,000 reactions. So if you put 32 organs together, this is the overall reactions that you're getting. Word of caution, do not download these models to your local machine. These are pretty huge. Try to do it on the web version. Okay, so there are high chances the machine gonna get hang. Okay, so just be careful. Okay, so these are the different adult organs. Yeah, so here we did not build everything from scratch. There were certain models that were published before the Harvey-Harvetta models. These are the small intestine. So we were very specific to pick only those previously published models which were built from scratch, or the bottom up reconstruction, the language that we used in our computational terms, where we use a lot of literature evidence to build these models. So we used previously published intestine, adipocyte, liver models, and these are pretty well established models, have been used over and over and over again. And that's how it also ended up in our system. And these are the blood cell types, and then as I said, we also had a lot of these gender specific organs. Okay, so we integrated them, we did a lot of tests. What did we finally achieve? One of the good point that came out of this was, now we were able to do a lot of these biofluid specific biomarker identification. We simulated close to 60 diseases, all of them metabolic diseases, and we were able to capture with high accuracy their biomarker prediction. So this is what you see, the comparison of the whole body models in capturing the biomarkers versus the previous generic model like the Recon. So this has got a lot of application when you use this particular model for biomarker identification. That is, if you want to go for gender specific study. Another interesting thing that came out of this was we were able to capture a lot of this organ-organ crosstalk, again at a metabolic level. One of the high point of this was, let's say you are perturbing one particular organ, how is it then connecting to the other organs and creating these systemic level clinical symptoms, okay? And that is most of the time is being answered by your metabolic crosstalks. One of the important pathways that we were able to achieve was, we achieved a lot. I just took one example which is called the glucose-alanine cycle, usually important for the skeletal muscle. Whenever we are exercising, we tend to use a lot of glucose oxidized to pyruvate. Not only are we releasing lactate, but we are also releasing alanine. These two products then through the blood enter the liver, which will then convert back to glucose using the gluconeogenesis pathway for glucose production, and then it gets back into the skeletal muscle. So this is the organ-organ crosstalk at the metabolite level. We had captured this for a lot of different organs, and we are in a position now to simulate a lot of different diseases where you see systemic level and lot of organ involvement in one particular disease. So this is one application of this particular large models. Another application I would like to show you is integration with the microbe data, the gut microbe specifically. So we have a lot of metadata for around 150 healthy individuals, and again the whole body models as such are pretty large. Now you are integrating it to a microbial reconstructions which are again pretty large. So the complexity of this particular models are pretty high. What we did end up with, we had these phyla or the class of microbes which are highly represented in the gut. We took those and we asked the model, are you able to capture? And it did, and what extra it showed was, these are the neurotransmitters, and there is something called the gut-brain axis. Now it is the gut-brain-kidney axis. So which means that the neurotransmitters are being synthesized by the microbes in the gut, which then has the capacity to associate with that of the brain functions in the human body. So which is kind of showing you the potential of this particular model to interrogate the brain-gut as well as the brain-gut-kidney axis. Okay. So these were all the important features that I want to discuss today with respect to, we started out with a lot of data. It could be clinical data, it could be omics data, it could be physiological data, bibliomic data, whatever data you take, you put that onto a model, you integrate it, and then you end up having these condition specific or disease specific models. It gives you a lot of mechanistic insights, typically at the level of the biochemical basis of the disease. And we are at a level of creating something of a virtual human, but the word of caution here is we are capturing only the metabolic aspect of the human. We are completely ignoring the non-metabolic functions. So have to be careful when you are interpreting the results. And then of course, when you combine the computational strategies with that of the wet lab experimental validation, it has got much more impact than doing it alone. So that was the different human metabolic networks out there, the different large scale models out there, and then all of this very much follows the lines of the modern medicine or the scientific wellness these days that we talk about, which is the predictive, preventive, personalized and participatory, or the P4 medicine approach. I just usually have this as my final concluding slide for majority of the presentations. I just truly believe in this. I hope I inspire you to read up this book, which is Scientific Wellness by Professor Lee Hood. He talks about all of these networks, disease networks, and how you can actually go from a population level to a much more personalized therapeutic strategy. So yeah, again, all of this was not done in isolation. There's entire teams involved, my collaborators involved, my students involved, and I was part of IIT Madras before joining Vantage. So most of the work that was done was in association with IIT Madras, and the funding was generously given by DST Inspire Fellowship. And of course my students, so, and then these are my collaborators. So yeah, thank you.
Thank you, Swagatika, for this wonderful talk. So we have time for 2 quick questions. Maybe I have a question. So what is the state of the art in terms of objective functions when you are doing flux balance or constraint-based modeling with these humans? Because with cancer, you can of course use maximization of biomass, but when you are doing the microbe,
under kind of cancer which is called the colon cancer. So duplication of the cell, of course, but there are various these non-growth supporting functions also which needs to be optimized for. So typically what we have done, we have again taken it from the microbial studies, which is called the Pareto optimality. You can do 2 objectives at a time, or you can do one after the other. For the cancer, what we did is we put one objective, but we had the other objectives as the constraints.
So how many objectives are possible in Agora directly?
At a time?
No. How many Agora to? I have not read the paper. I'm sorry. How many different objectives, possible objectives, Agora 2 gives away?
So Agora currently has close to 800 microbial reconstructions, and I would say each going for at least one biomass, so 800 reactions, objectives.
Thanks. Questions?
Thank you. It's a nice presentation. Just for my understanding, like the role of microbes involved in the, like we give an example of neurotransmitter, which always regulate the human metabolic regulation. So what will be your opinion, like level of contribution of gut microbiome? Even always like it is a second genome of the host always. So what is a response? Or those are the key source or like for the regulation of gut microbiome in that like kind of host metabolic pathway, particularly the human metabolic pathway. So you are mentioning about the neurotransmitters. Can you just brief or something that is a key, or the human metabolic system also produce the signals, or microbes are the key signals?
Both.
Yeah.
Both way. It is both ways. So neurotransmitters, because I am interested in brain-gut axis, that's how I showed this example. But microbial has got lot of interactions not only with the intestinal region, which is the large intestine where you have the majority of your microbes, but it starts from the level of oral microbiome to the level of stomach, small intestine, large intestine. So gut microbial information, of course, important, but these days it is just going beyond gut as well. There's microbes on your skin. There's we thought that when a woman is carrying an infant or a baby, it is most of a sterile environment, but it's not the case. You also have microbes in that particular sterile, so it's not sterile anymore. So you cannot actually escape microbes And gut microbes is just, we know more about it, but there's various other kinds of microbes sitting on the human body yet to be explored.
I think we can thank Swagatika for this wonderful talk.